No, it would be very visceral, you know. Hi. So, this morning as I was going through my email, my colleague Doug Rosine sent me this paper. So he's on a study section looking at Alzheimer's grants, and one of the grants referenced this manuscript that he downloaded. And so it's an accepted manuscript on the origin of the vertebrate body plan. And they've got all these embryos laid out like this. So I was just very jazzed. What these guys are saying is expressing how the complex embryos establish their shape, go from a small group of cells to an organism by a series of geometric transformations via a series of expanding and contracting and multiplying three-dimensional stretched rectangular grids. I feel if Einstein is watching from someplace, he'd be having a good day. This really is this Einstein really did set a lot of things in motion. Who knows about the Einstein to cubism connection, where the cubists said, those physicists should not be the only people who should be authorized to mess with space and time. We can do it too. And so they started refracting things. As I mentioned, D'Arcy Thompson yesterday. Well, let's assume for a minute that I have your full cognitive attention for 30 seconds before the torpor of lectures overtakes us. So this hypothesis about the grid structure of the brain really is run by a single mathematical theorem. Now, I can give you a lot of names for the theorem, but the thing about really important theorems is that you can't get around them. Getting around them requires a whole new big discovery. Now, this theorem, let's see, it goes by many names. That sounds like a joke. It's sometimes called the Frobenius theorem, but actually, everybody knows that that's a misnomer because Frobenius proposed it, but he didn't prove it. So that in math, that doesn't get you to name it. The people who proved it were Dehaene and Klebsch, the guy from the Klebsch-Gordon coefficients. So it should have their name. But since then, it's been called things like the fundamental theorem of partial differential equations and also the fundamental theorem of differential topology. And it's been generalized far beyond its original framework. Its original statement was in geometry. People said, how come we can solve these ordinary differential equations and we can't seem to solve these partial differential equations? Are they just trickier, or is there something more fundamental going on? And indeed, there was something more fundamental going on, and they got it sorted out. And hence, it's the cornerstone of a field. The field of which it's a cornerstone is called differential topology. At least it used to be called differential topology. It was a field that kind of came into existence in the 60s when, I don't have to describe the history, but it has had a very successful career and now is extremely important in field theory. So the people who do things like quantum gravity or gravity theory use this, kind of get out of bed and have this with their cornflakes. This theorem was given as a basic one-line exercise in Meisner, Thorne, and Wheeler's gravity book. Guys who all know who Kip Thorne is? Anybody who know Kip Thorne? So Kip Thorne, Mark does. Kip Thorne has become a culturally important figure. He's old now, but it's better than the alternatives. But he's been doing gravity theory for 50 years, and he's the guy who he came to prominence in this town because he was instrumental in getting that movie Interstellar to work right, getting those black hole simulations to really look reasonable and to show the important stuff. He's the guy who really pulled together the gravity wave detector. So there had been a lot of ideas for gravity wave detectors, and basically the people who were trying to put together, they had built many gravity detectors since Weber built an aluminum cylinder in the 60s that was like 8 feet across and it like a bank vault door. It didn't work. And so can you imagine trying to sell to the NSF, we really need you to write a check for many $billion with a B to do something that we've tried to do 50 times and failed for the last 30 years. And we've really never even seen any of the objects that might produce a source of the kind we're describing. And yeah, we're trying to measure at 10 to the minus 21 meter displacement over a baseline of 8 kilometers. So we're going to put in 100 kilowatts of lasers that are going to go 200 times up and back in a tunnel, and we're going to factor out other sources of error, like the gravitational field due to sparrows flying overhead. We're going to get rid of that stuff by processing, and it's all going to work. And so he was the guy who kept that project on track. He didn't, he was a theorist. Anyway, oops, wrong side. So there's a symmetry in higher dimensions that it first occurs in 3 dimensions. It doesn't have a 2-dimensional counterpart. You'll see why in a second. But it's really a rather simple idea. So, and I'm so glad that I have 2-color markers. Okay, what is a differential equation? So the simplest way to put it is, imagine that you're given a vector field, a set of lines, let's say, in 2 dimensions, whatever. And then what a differential equation is construct families of curves whose tangents these lines are. Okay? Now that's an ordinary differential equation. And the typical case is something like the velocity of water in a stream. So you have a stream flowing by, and you could put a Doppler machine and measure the velocity at every point, or a little flag in the water, and see which way a windsock, and see which way the wind is pointing. And then you could throw dust into the water. And that dust has to go somewhere, whether the water is compressible or incompressible, whether it's turbulent or steady. The dust is going to go somewhere. Actually, in this case, I'm talking about a time-independent velocity field. So in a real stream, these velocities would be changing. But you can imagine a steady one, or even the time-dependent one. And then the fundamental theorem of ordinary differential equations is you can always find the curves. The curves always exist, and the curves are as smooth as the vectors are. What does it mean to say the vectors are smooth? What it means is the vectors change gradually. So as you inch your way from point to point to point, the vectors are not doing, and the next point is this. That wouldn't be continuous, because you moved a tiny amount and the thing suddenly jumped. So they can go to 0, but okay, that's what an ordinary differential equation is. And you can always solve them. By solve, I mean the solution exists. That doesn't mean you can write a formal solution all the time. It just means that the solution exists. And there are these curves. And by the way, they can do all kinds of things. So the curves, you could set up a field you could set up the circular field, right? So the vectors could, the rotation vector field and it's continuous at 0 because the vectors are getting smaller. The velocity, as you get to the center of the circle, the velocities get littler. Here you have this velocity here, then you have this velocity here, this velocity here. So it's still continuous because they're getting little, 0. And this is rotation, and the curves of this are this. Ordinary differential equations. Next step up. Partial differential equations, ugly words, but simple idea. Suppose somebody gave you a little, not a field of vectors anymore, but a field of little 2-dimensional planes, little playing cards. Okay, so a 2-dimensional plane field in 3 dimensions, let's say. It only gets interesting if you're in 3 or more dimensions. So you have these little planes filling a volume. And there's a definition of, are they continuous? In other words, when you move from point to point, the orientation of the planes changes gradually. Now, how might such a plane field arise? Why are we talking about it here? Well, suppose you have 2 sets of fibers in a volume, like 2 crossing fiber pathways in the brain. Then, at every crossing point, there are going to be 2 vectors, right? So those 2 vectors define a little plane at that point. In fact, there could be a lot of different fiber fields that define the same plane. It could be these 2 vectors, or these 2 vectors. They don't have to be at 90 degrees. or they don't have to be in any orientation. They're all basic, they're all defining the very same plane at this location. And so you fill the volume with a set of planes. Yes? No? Okay. So you got, it's a funny picture. How would you actually build such a thing? There are liquid crystals that will do it. You know, there are little liquid crystal molecules that are shaped like little pancakes. You know, we tend to think of liquid crystals being shaped like little worms, nematic, you know, worm shapes, but they're pancake crystals. They're called smectic, after the word soap. I don't know if soap is really smectic. I can't remember. Anyway, you fill a volume with them, and they're going to try to kind of stack together in little planes, little local planes. So it's the same question. Given a set of local tiny planes, little microplanes filling a volume, can you find continuous extended surfaces that touch all the little planes tangent? It's just like the case we asked in the very first instance of the lines, curves, ordinary differential equations, next dimension up, higher dimension, little planes. Are there or are there not surfaces that connect the planes? Any takers? It's not intuitive. The answer is, in general, no. Those surfaces, in general, do not exist. That's why people couldn't solve the partial differential equations. It's not that, let me explain why. The reason is really simple. I have many, many slides of this. None of them are adequate. The reason is that I can arrange these four planes like dominoes, like an Escher staircase. Let me do it by hand before I show you the pictures. You know the neuroscience of this. If you show somebody, if you read something, you're engaging more abstract intellectual faculties than if you see a movie of it, right? You get all this concreteness that is irrelevant to the message. So that's why I'm so painfully talking my way through this instead of showing you a picture which you would see but not necessarily understand. And trust me, this is an idea that, you know, everybody in the math department here who does differential geometry, who does geometry, I mean, anybody who's a professor of math here, this is the kind of thing that would be on a standard PhD qualifying exam in any math department. And a physics department if you're doing related physics. So this is all very well known. Let me explain the problem. It's quite simply this. Like an Escher staircase, if you have, you need to consider, well, let's say 4 planes. So imagine like the Escher staircase going up, 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 up. Here's the question. Let me put it to you. Let me prove the theorem actually. Suppose there were a surface. Suppose that there were surfaces in this space. Okay? Well then, and they're tangent to these little planes. Then I could walk in this direction and stay on the surface. Then I could switch to the other vector. Can you see me? I could walk along one vector field and go in this direction. Then I could switch and I could walk over here. Then I could switch back to the first one and go negative x, and then I could come back here. And when I come back to the starting place, I better be at the same level. If I've shifted levels, then there was no way to fill in that this loop I couldn't fill in with a single surface. And so what I'm really saying is, if you think of the 4 lines that are the edge of this thing, I need, well, 4 straight lines have to come back to the origin, have to come back to the 0 height. Now, what's the probability that they do that? 0. Because there's a lot of ways to be at something different. All I need is one of these segments to be out of kilter. So I need these 2 segments and these 2 segments to be in some kind of a balance. So there's an extra condition. The partial differential equation case the cool theorem is that's the only thing that stops partial differential equations from being solved. So if you stay in a sheet, if you can run these little test curves and you don't travel up or down, then there is a solution. And if that doesn't happen, then there is no solution. Now, another thing that's a little hard to believe is that this actually is defined even at a single point. I can make that loop as tiny as I like, and I will still find a tiny little disparity. So it's not just a finite loop. It's even trying to build a tiny solution is impossible if these things are out of kilter. So there was a question yesterday. This is what we've been kind of working around towards. Oh, there's one other thing you might want to know. What does having, suppose you have little microsurfaces and you can slice the space into a set of sheets, okay, like an onion. Now, what do those sheets entitle you to do? What are they equivalent to? They're equivalent to a function. You've now got floors in a building, and every floor has a floor number associated with it. Which floor are you on? In other words, they are the level sets of a function, which we can call height. Which layer are you in? The question of which layer are you in is well defined. If you can't integrate the plane field, then there really is no definition of height. It's like those parking garages. You ever have the problem of being lost in a parking garage and saying, am I on level 4 or am I on level 3? And you start out on level 4 and you keep walking level, 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 but at some point you've gone from level 4 to level 3, and there was no bright line on the floor to tell you when that happened. You're just sort of magically there, because it actually doesn't have well-defined floors. The difference is that the spiral ramp in a parking garage is not really as bad as this, because it's actually just got this singularity. It's just got a spiral. So if you just black out the spiral, it's really one. But actually, let me give it to you another way. Let me give you this in yet one more point. Suppose I wanted to introduce a function into this volume that would have different values that would tell different values on every space. Suppose I could crawl from one level to another. Well, then I could get from any point in the space to any other point in the space just along curves. Because I just go in a little spiral over here, crank my way up to another level, and then I go across. So any function that's constant along the curves has to be constant everywhere. There's no useful, okay, why the long spiel? Because this is how embryology worked. Because embryology located things in space using functions. What were those functions? Concentrations of molecules, concentration gradients. So they were, you have an organizing, you have a pair of molecules, let's say, epherin A and epherin B over here, and position between these two organizing centers is established by the relative ratio of, by the intensity or ratio of those two molecules. I'm sorry, why did I trip over the word ratio? There's something very cool going on there that I will share with you. So many molecules that organize position in the body actually don't use the absolute chemical concentration of the guidance molecule. They use the ratio of a pair of guidance molecules. Okay? So they'll use, you'll have one guidance molecule on the left, the opposite molecule on the right, and position between the two will be not the size of either one, but the ratio between the two. Can anybody think of a good reason why you would use ratios of guidance molecules instead of individual amplitudes? Anybody? Mark, go ahead. And how does that help? That's essentially, that's correct. Another way to say it is that suppose the animal gets a lot bigger. Suppose it stretches like heck. So you start out with an embryo that's this big, and you want to say, here's the left eye, here's the right eye, and you want to have shapes defined in this embryo, but then it's going to turn into an animal that's this big. It's going to inflate by a lot. So absolute concentrations will just drop, but ratios will stay the same as the thing stretches. So there's stretch invariant. So you can just build the thing like this and stretch it. Okay, so the question was asked yesterday, how do we know when diffusion tracks might be right? Is there anything we can do to tell, based on the appearance of the data itself, really? I mean, do we have a criteria that tells us when it's right? Now, and I tried to you're really coming back to the central question that I was discussing yesterday, which is how do you know, or how might you know? Now, the how should you know question would be, well, if we did, so if we had a gold standard, so one way to do it would be just brute force. You say, okay, here's a gold standard brain whose microscopic and macroscopic structure we've mapped. So we've done maps down to a nanometer scale up to the whole brain, and we know the orientation distribution function for every single voxel, and so we try to build an inference engine, something like that. I don't know. It's sort of the validation question. But I think the question was, you really wanted something simpler than that. You didn't want to just say brute force. We know because we actually found it out from some other thing, and all we can hope to do is check how close we are to something else. Certainly a method that had that ability only would be of no value in looking at human variation, or in fact, looking at live humans, or looking at cases that were not yet understood. So for example, you have a genetic variant, or you have a disease process. You want something that will be robust in all those cases. So your question was, how do you know if it's right or not? And indeed, that was the sticking point. That was the place where basically the situation seemed hopeless. And really, on its own terms, it would be hopeless. If you say, how do you tell, given that ordinary differential equations, where would you have a constraint from? You see, okay, let me give you a situation that wasn't hopeless. Here's a nice situation. So as Mark mentioned, I did MR angiography back in the 80s. Well, in angiography, there turns out to be a kind of test for whether things might be real or not. So given a flow velocity field, what can you do with it to tell if it might be a candidate for real flow or isn't? Well, real flow is incompressible. And it conserves volume. So you've got a conservation law for real flows. If you're just seeing stuff, if you're just seeing signal, brightness in a vessel, that's one thing. But if you're measuring the velocity, you can kind of tell if it's right. Because if you measure the velocity, if here's a pipe and you measure the velocities here and you add them up, and then you better measure them here and add them up and get the same number, or else there's something wrong. Or else you've got a leak in your pipe or a leak in your methodology, which is more likely. So more extensively, you could even say, and I guess this was your question, do velocities have to satisfy a Navier-Stokes equation. So in principle, if you had a really accurate velocity field, you could compare it to the physics and say, does the physics, the heart's pushing on this side and the vessels are shaped like this and they're bouncing off they're elastic, would this velocity field result? You could test it because you've got a theory. You've got Newtonian mechanics that tells you how flow is supposed to be shaped given certain forces and given the properties of the liquid, right? Now, do we have a theory like that of the brain? Do we have a theory of how nerve fibers are supposed to conduct themselves from one place to another? Not exactly. We have developmental and embryological ideas, and then we have our ideas, and they're kind of ad hoc. So anyway, long story short, we were looking around and flat out stumbled on this principle of crossing fibers forming sheets. And given that it was rare a priori, we had to accept it. We had to say, this was something that even though we didn't know that it was happening beforehand, it was not undeniable once it was observed. So let me show you, let me try to show you this. Let's see, we need the projector back on. All right. And can we drop the room lights on the front? So this is one picture of what we were talking about. The general case is on the left, where things don't line up. And what we're saying is that we happened to find in the brain that things do line up. And this planarity of orientations makes sense from a variety of points of view. Here's another way to see what's going on. you have horizontal fibers and vertical fibers. And the horizontal fibers and vertical fibers might be related to each other in a lot of different ways. There's a lot of latitude for how you can tilt. I mean, for example, here the horizontal fibers are, spiraling around. And for any spiral pitch except one, there are no mutual planes between horizontal guys and vertical guys. There's no such thing as a surface common to both, in which both track in which multiple fibers of each run in the same surface. Okay, nothing except in that one case where they're matched. So what we're suggesting is that the pathways of the brain have this matching property. Now, to better capture this matching property, we devised a very simple algorithm where here we have blue fibers running in one direction and purple fibers running in another direction. And we just measure the gap between the two. We measure that offset gap and keep the stuff where the offset in the z-axis is small, where the offset is below a threshold. So if they're not in the same two-dimensional surface, you'll get a very small object. But if they're tracking together, if they are in the same surface, then you get this large structure built entirely of pathways. So this is a very handy way to visualize whether this process is occurring in a given data set. I'll come back to this in a minute, but I wanted to just kind of get them out. The slide is out of order in a sense, but there's no linear order to present this. So anyway, here's a typical picture of pathways of the brain. This is just DSI in a normal volunteer. But what's been done is that we've just selected fibers of the internal capsule and fibers of the SLF fascicle. So internal capsule is gold and green. Lateral fasciculus is purple. And you can look at them qualitatively and see that they look like a woven sheet. So this tendency to form two-dimensional sheets is what we're talking about. Now, this is a rhesus monkey, and these are simply selected paths in the volume of the sagittal stratum. So the sagittal stratum is a visual association hunk of white matter located in the mostly in the occipital lobe, kind of underneath the occipito-parietal operculum, and you got paths running in three directions. You have to stare at this for a minute. Some people have very different abilities to see stereo or see three-dimensional figures from 2D things. You've got a front surface. So you've got two surfaces defined by these beads. So you got these beads in the corners. And then in white are the fibers that go through the beads. And then in color are the fibers that go through those fibers. And so through this bead, you got fibers in three directions. These are just all the different fibers that you pick up in this bead. And through this yellow bead, there are fibers in three directions. Well, actually two. This surface this way, surface this way. Actually, the yellow one is the back plane. The blue one is in the front. It's very hard to see that in this, I agree. So these white guys meet. They're tracking in a surface. That's the tracking in a surface principle that I was describing. So maybe you can see it now, that these are meeting here and here. Look at it this way. So here's this guy, here's orange, here's yellow, and here are the white fibers coming out of them. Now, I could potentially adjust the relative position to make one or the other meet. But having both of them meet at the same time, which is the key requirement for this test, that's the thing that there's no way to force from the outside. It either happened or it didn't happen. So the fact that something odd is happening widely is a test. It's saying the fibers, in effect, have to be of a certain level of accuracy in order for this to be manifest. If they were totally random or junk, you would never have a pattern like this visible. It's a little bit like saying, how do you know your eyeglasses are working well, or how do you know the eyeglasses are successful? Well, you write on a piece of paper the combination to a lock, and if the person can unlock the lock by correctly reading the numbers, then you say, okay, glasses are okay, because there'd be no way they could unlock the lock unless their glasses worked. Same argument, really. Even though, and here's the funny part, even though we didn't know that this was the case beforehand, that was the point. It made no difference that we didn't know that fibers in the brain were like this a priori. It's that given the observation, there's no escaping it. So anyway, this illustrates that this is happening not just in one sheet, but in multiple parallel sheets in this volume. I'm going to call your attention to this. I don't remember which position I put this slide in. There's a very funny tracer injection that I found in my friend Jeremy Schmauman's book. Actually, you know what? I'm going to go to all the slides and see if I can find that tracer injection just right now, because it's so appropriate. Except my slides are going to be slow to load. Do I have it? Yes, here we are. Back to, oh gosh, yes. Yeah, here's the sagittal stratum again. So Jeremy did a tracer injection. This is in Schmauman and Pandya. And just appreciate this for just a second, because this is work from the 90s published in the early of fiber pathways of the brain. And this is what they found circa 2000. Tracer injection in temporal lobe, which then goes inward. I'm sorry, take a look at that. Do you see what it is? This is exactly what we were looking at 1 second ago. This is not a curve. And then it goes bang, bang. And this is that extraction of the crossing paths that I showed to you. So this is, I'll go back and show more of these images. But this is the surface of the sagittal stratum here, the deeper plane of the sagittal stratum, grids in this axis, grids in the coronal axis. So this is a sagittal grid that goes in, this is a coronal grid in blue. These are axial grids. And indeed, the grid structure strangely matches this tracer injection. So it looks like, as Carl Zielis said, we already knew this. I don't know. And here's the confocal microscopy of the same area showing fibers in 3 mutually orthogonal, close to orthogonal directions, 3 independent directions. So I needed to digress to show you this pattern of is it a sheet, how sheety is the data, so that you would be able to spot it and recognize that this surface structure, to notice it when it occurs. If you don't understand, if you can't see it or understand why it might be significant, then I'm not doing my job in the sense of I'm just telling you something to take on authority rather than equipping you to critically evaluate the question. Okay, so I'm trying to get this thing back onto the screen. So this is at the level of the stuff in the science paper that's from 2012, this is sort of what we were doing. Pick a point in red and find a sheet of fibers that goes through, find the fibers that go through the point in white, then find the fibers that go through those fibers. Does it look like a sheet? So look like is not a very strong test. Is bound to cause trouble. The first, so here's what the fibers look like. They certainly do look like a sheet. Now remember that from the standards, let me be quite so insulting about qualitative analysis. Qualitative analysis is the only tool that track tracer studies or that neuroanatomy had from the 15th century to 2000. The move toward quantitation has been the result of our success with digital computing over the very recent decades. In any case, here's what we see. And this is really what prepared me to see this. This is the heart over on the left. And the heart very strongly, here I have 2 directions. The yellow are the muscle fibers, and the red are cracks between the fibers. The heart is a very strongly twisted structure. So this property we can call twistedness. And what we're saying is the heart has twistedness. The brain does not have twistedness because these things line back up. That's this line-up property. We can call it twistedness. It was called that by the late Bill Thurston. The point is, the heart doesn't have it, so the heart will not fall apart like an onion. Having a heart that falls apart like an onion would be a bad thing. Specifically, you want forces in the heart to distribute in 3 dimensions, hence its rope-like character, its twistedness, and the fact that you have to cook it for 2 days. But the brain is different. And of course we don't know why, but it's got, so what's the other point? There's something else going on here that I'm not being clear enough about. Remember, we have a sheet here. Now, I said earlier, it wouldn't have mattered how these other fibers were oriented to make it sheet-like, right? They could meet at 90 degrees, they could meet at 20 degrees, they could be straight, but they also could be very twisty and turny. Why aren't they twisty and turny? Because this grid appearance, what's significant in this data is the sheet in this direction, but what's also going on is the grid structure signifies sheets in the other 2 perpendicular axes. So it's not just one set of sheets, it's 3 sets of sheets, xy, xz, yz. Anyway, here's what I was describing to you a minute ago. So here we try to get more quantitative. Here we say we have an objective method to identify the fibers that cross to the limit of our digital resolution. So the red stuff and the blue stuff, that's the stuff we saw qualitatively back when. And the white stuff is stuff that has been measured to fit the sheet condition to within 1 voxel, in other words, to the limit of our digital resolution. And so you can now, this little algorithm can then be applied to an entire data set. And you can say how much sheet structure is there in this entire data set from top to bottom, where is it located, where is it not located, how many of the how many of the orientation vectors are not absorbed or into a sheet structure. So how much structure, how much diffusion information is there that is not sheet-like? So you can ask all these questions once, now that we have an objective algorithm. We knew that we needed one. It was good to make one. Anyway, just to show some structure, here's human brain. The white are sort of the traditional pathways, and the colors are sheet structure that was detected within this data. I think you understand in the program that the program spits out lots more stuff than you can see at once. The sheet structure is much the same. So the program generates gazillions of these grids, and then you have to winnow them down in order to be able to see them. And the good principle of winnowing is big ones are more interesting, more significant, tend to be more useful, but they don't tell the whole story. Here's rhesus monkey. Same. I'm sorry. What am I showing? Let me talk about this for a second. So this is the callosum. This is a human. We're in the coronal plane. We're looking down the middle like this. The callosum is a big arch. It has fibers running both left in its perpendicular thickness, which certainly have been seen under microscopy. This is the cingulum bundle, and the cingulum bundle is perpendicular. So it's parked on the callosum. It's the z-axis. So they have the front to back axis in common. The callosum is front to back and mostly left to right. The cingulum bundle is front to back and in. So think of it as perpendicular to the callosum. And we see the same thing in other species. Why is this interesting? The question is, what are gyri like? There's a real question here. What is a gyrus like? I mean, you got all these funny gyri on the top. So we're demonstrating, building up this grid structure in the middle of the white matter, in the centrum semiovale, in the corticospinal tract, in these big bundles. But how do those relate to the gyri, which are all curvy and sticking out? Well, here, it seems in this instance, they seem to relate very harmoniously. They have one of their 3 axes in common. The front to back axis is common to both, and the other 2 axes are logical in each other's terms. So it's the same, it looks like the same coordinate system, just different parts of it. You know, one plane, you know, an XY plane and an XZ plane, if you like. So interesting that that might be what other gyri are like as well. Here's the monkey, and for some reason these little green inset images are not appearing at all. They're just slow to load. Yeah, this is you know, a cingulum bundle in which we did some histology, but you see very similar structure as we did in the human. I'm not going to wait. If I have the option to not wait. So, as I was saying about TrackViz, you know, here is a potpourri of large grid structures. This is a rhesus monkey brain, and I must emphasize again that these structures are merely tractography which has been, of which a subset is selected based on the condition of making two-dimensional sheets with their neighbors. So there's no build a sheet, look for a sheet, build a grid, look for a grid. It's just these are the fibers trimmed and selected based on are they partnered with another direction. So I don't know where I should stand and discuss this. We're looking at the, so here's cerebellum. Pons is a very nice three-dimensional grid with planes in all three axes. Sagittal stratum that we discussed a moment ago. Ventral frontal, what is that? That's above the Sylvian fissure. This Muratov bundle runs over the dome of the cortex, over the dome of the caudate nucleus. So you have callosum. Shall I go through this again? So we have centrum semiovale, which is really a three-dimensional, a triangular prism. It's basically a grid that goes through the callosum and then just fans laterally. And so the thing is a three-dimensional prism-shaped box with a short side toward the callosum and a long side away from the callosum. What are other structures? So let me spin it again. And in a few instances, I've drawn in a few popular pathways just to give some frame of reference. So here's the cingulum bundle. And it's interesting that these beautiful geometric curves of the cingulum bundle don't seem to have a grid that goes with them. Probably they're an artifact. Probably what's really happening is that the fibers are actually making sharp 90-degree turns from the front-back direction to a left-right direction in the callosum. And somehow we smush those together due to spatial and diffusion resolution and come up with this smooth diagonal. It's probably the case. Let me just run it again so you can enjoy the show. So you have centrum semiovale on both sides. The callosum, so the callosum is actually one big rectangular box with the centrum semiovale. And so what happens in the front? The forceps just is this callosal box bending down 90 degrees and then 180 degrees, all the way down and around the front. In the back, it expands out into the sagittal stratum. In the top, it arcs over medially and then down again. So you can begin to comprehend the entire structure of the deep white matter of the brain as just a single highly curved grid structure whose pieces we are now able to identify. So here we did a little test. Jeremy Schmann's book says in the rhesus monkey there are something like 7 major association pathways: SLF 1, 2, 3, ILF, MDLF, cingulum bundle. And so we wanted to see whether they were aspects of a grid structure, whether they had grid-like neighborhoods. And indeed, the only pathway that he couldn't identify, that we couldn't find in the MRI data, was the pathway, if you will, that he had called the extreme capsule. And he said he called that a pathway. The uncinate fascicle also gives us heartburn. I don't really understand the architecture of the uncinate fascicle running from the it's so twisting. But the extreme capsule had a very interesting resolution, which is that we couldn't see it in the rhesus monkey, but we could see it in the human. And in the human, it looks like a pie. It's a grid, but it's shaped radially and circumferentially. So I don't really understand the overall architecture of the brain, but probably the best way to think of the brain is that the amygdala stayed put and the brain spun 180 degrees around the amygdala. That it's kind of an expansion process like this. And if you think of the caudate nucleus and the shape of the internal capsule, it's not a bad idea. In any case, we could see it in the human. It was grid-like. So what's our problem right now? We have these grid-like neighborhoods, but we're still not able to uniquely glue them all together. So we have local grid structure, but we're a little bit stuck on understanding how seamlessly these things may weld together, and in particular, whether there are any parts that don't weld. If you look at your thumbprints, you will see the danger. So if you look at your fingerprint, you know that there are singularities. There will be little corner points or loop points where the concept of a coordinate system is really not working very well, where you have inconsistent coordinate assignment in a hairpin turn or in a place where three areas come together in a triangular configuration. We don't know for sure if any of those things happen in the brain. They very well may. So until we have a complete GPS map, complete longitude, latitude, and depth map of the brain, we will not be able to know if those things occur. Now, personally, I'd be delighted if we could find them, because if we could, they would be just like a fingerprint. Well, just like a fingerprint. You want to reduce the data. So if you, in a sense, you'd be able to reduce all this data to perhaps just a little bit of data relating to where those singularities are located and characterize everything else by filling it in. So there's an interesting message here. Now, by the way, I just want to step back from this. There are many ways to step back from this. Why do we have to buy expensive scanners? Because we want to capture more degrees of freedom of the brain, right? Resolution. So it's basically resolution per unit time per dollar. Now, what are we saying about the total number of degrees of freedom of cerebral white matter? We're saying the total degrees of freedom is much less than it looks. There is a strong constraint or interrelatedness within the white matter that causes one structure to be correlated to another structure. That means that once we mastered these ideas in this anatomy, we might be in a position to capture this structure at much lower cost with much lower technology much faster, because it's got so few degrees of freedom. We may need the million voxel image with 0.7 millimeter resolution and 500 diffusion measurements to discover it. But having discovered it, it might then turn out that you only need 1/10 that much data to see its variability from individual to individual, because it's so patterned. So, I mean, that's the kind of thing that Mark loves thinking about. How much data is there really, and how do you what are the biological variables that are driving it? Well, anyway, I've talked a lot. Here's back to the centrum semiovale. Let's see. I'm getting a little discombobulated. Yeah, so this is the centrum semiovale, and you see there are three directions. Now, here we've applied that automated sheet detector, and this really will totally baffle you three-dimensionally. So we have the Muratov bundle here. This is running over the dome of the caudate in red, and so this is an axial grid running like so. This green thing is a coronal grid. This is a fan of fibers emerging from the callosum and fanning up, as is the gold stuff, to the motor cortex, because we're in the precentral gyrus. And this purple thing, here's the one that's hard to see. This is actually parallel to one ring of the fan. It's like the top of a cylinder running in and out. So this is a tilted, this is the projection direction of the projection pathways that go, down. And so they are forming a cylinder, that's the surface of a cylinder, and this thing is going back. So the key is to look at this milky area and say, that's behind this slice. So you recognize that as going back, then you can see the three-dimensional shape. So this makes good on my suggestion that the centrum semiovale is a box, is a prism or box. And here's just some MRI in rhesus and in rhesus on the top and human on the bottom, I think. Here's Doug and Farzad's paper submitted to count fibers in specific directions. And as I indicated, you really have to do it with multiple tracers because you need to know connectivity. Is it really the motor efference that we are seeing, of which we have a representation? Is it truly motor efference that this is representative of? And then second, is that signal fully driving the MRI signal? So is that signal congruent with all of the fibers in the volume? Because water feels everything. So even if the motor fibers are like lined up in some way, unless they are lined up in the same way as the average, we won't be reflecting it. So you had to check it in these two different ways. This is another way to look at putting the pieces together. Here we've just picked out grid neighborhoods along a callosal fiber running left to right from motor cortex to motor cortex. And you have a little grid zone in the cortex, and then a three-dimensional grid in the gyrus, and association paths, and then the centrum semiovale, and then the callosum with some cingulum stuff, and then the same stuff going out the other side in the reverse order. So you see where we're at. It seems extremely reasonable to suppose that these pieces can be put together, but coming up, right now we're trying to come up with a machine learning approach that will allow us to quantify how well we put them together. It doesn't do any good to just force them to go together and say, okay, here's the solution. You have to have a measurement that says how consistently do they fit. That's really the trouble. If you don't have an error bar on your data, it's very easy to end up in deep water. So the real problem is the error bar. How coherently did these do the pieces fit together? But how many people have played with Legos? Yeah, okay. Here, this is a close-up. This is a human, we looked at the U fibers between the frontal and prefrontal gyrus, central and the precentral and, let's see, we see motor and premotor cortex. So the U fibers are in white, and we have a series of grid neighborhoods. So yes, this structure is not limited to white matter. But white matter is just an illusion anyway. It's just a specialized form of stuff. If you look at a bird, there is no white matter to speak of. There's just mixed tissue like the striatum in a rat. The quadruputamen of a rat is just kind of mixed up white matter pathways and gray matter pathways. Some people have said we're just picking up 90-degree angles. That's plainly not true. Here we've color-coded the angle between these fibers, and the angles change. Now, is this an interesting signal? Yeah, sure it is. It's real interesting. What changed the angles? Shearing during development. So something had to undergo a plastic deformation from a default orthogonal case. So there is a sense in which mapping these angles is showing you how growth happened in the developing brain. It in a way is like a retrospectoscope. How much deformation was needed to achieve the final state? Now here's another interesting image pair. The suggestion was made by some people that maybe this was just an artifact of low resolution. Well, it's a complete non sequitur because low resolution has nothing to do with two-dimensional planes. But in any case, here's a simulation of two B values in the rhesus monkey, 25,000 on the left and 40,000 on the right, and how much grid stuff we reconstructed. And so you see that this method doesn't appear to fall prey very strongly to Pierre-Pauli's concern that better scanning produces a more complete view of the grids. It isn't that it changes, it's that you see more. So that's kind of interesting. It sounds like this structure could be used as a metric for how good your diffusion scanning is. You see a lot of grids, you've done it right over more places. You see less, you see less. I should have packed the slide where we compare the connectome scanner at MGH to the WashU standard connectome scan. And their scans contain grid structure in the centrum semio valley and a little bit else. And ours, as you see, are like filling out the gyri and and starting to see gray matter. So better for now is plainly better until we figure out how to do better easier. See, here's something else that you analysts might appreciate. Here we've just counted the number of conventional paths and the number of sheets or grids at 2 different B values. So let's assume that higher B value is higher resolution. I mean, that's just engineering. So higher B value equals higher diffusion resolution. So the distribution and density of conventional track solutions didn't seem to care very much as we doubled the resolution. When we went from low res to high res, we kind of saw the overall number of tracks didn't dramatically change. But the distribution of the sheets changed dramatically. We were seeing a lot more stuff. So my suggestion is this is, you see how it all fits together. It looks like the suggestion that the sheet structure could be a metric for a threshold accuracy of filling in these fibers remains consistent. This is just a really high resolution scan. Here we took a rhesus hippocampus and scanned it at, or temporal lobe, and scanned it at 250 microns. This was 9.4 T scanned for about 4 days. Needless to say, there was not a monkey wrapped around its hippocampus. And here I've edited out the fibers, but what's really interesting, the first structure that I ever saw in the brain that looked very grid-like, and this was over 15 years ago, was the alveus of the hippocampus. The alveus of the hippocampus looks exactly like Chinese finger cuffs. It's just got 2 counterwound spirals of fiber orientations. Now, you can't generalize from that structure. It's such an oddball. This bundle of cerebral white matter running on the surface of the brain. It's just a very atypical kind of structure. So you shrug your shoulders and go, could this be generally the case? What was interesting was that we find in this image within the cortex, grid structure arranged in what looked to be like cornrows. And you see this in the hippocampus, and you see this in TE1, which is one of the temporal visual association areas right on the bottom of the temporal lobe. And the fibers look like cornrows. And I'm wondering, is this real? It's right on the hairy edge of our spatial resolution, but we've seen it many times in different settings. And just last week, I got an email from Roger Tutel, who showed me some microscopy of the visual cortex in which the neurons were indeed in rows. There was a clear periodicity of order of the neuron versus fiber density as you marched rostrally. So the question, we're very accustomed to thinking of neural structure of the cortex in terms of columns. So we say, okay, it's made of columns, but we don't think too much about whether the columns have much periodicity. Except the entorhinal cortex, there aren't a lot of people who would say the columns would have much crystalline structure, much periodicity or regularity. But I think we're seeing a lot of cornrows. And so it's an interesting question whether rows of columns or just rows of neurons is a ubiquitous finding. I think there's some interesting evidence that it may be. Well, this was a slide that I skipped forward to earlier. This is just the ponds. Now, this is just a total mess of gray matter and white matter. And there's like a zillion nuclei in there, right? I mean, there's like all kinds of stuff that we're looking at. And yet, when we do this MR diffusion averaging process, these are the average directions that diffusion MRI detects to the best of our ability. Another cute joke was, this one. The people in science called me up and said, okay, we really want a great cover. So I said, great, I'll make you a cover. But underneath, so this looks like relatively orthodox tractography. But the identical data, when you actually reconstruct where are these two-dimensional surfaces or sheets, these are the same lines. This is the same underlying NMR data turned into tracks by the same differential equation. The only difference is here we've organized them as 2D sheets. And so, and this is a normal human subject scanned for half an hour in the, connectome scanner, about which I've said not much. Well, I have no idea. Well, there are too many ideas as to why these sheets or grids might be present. As I was saying to some people after the lecture yesterday, if you ask why is an eyeball round, there are many reasons. One, it's easy to build a round ball. Two, round balls swivel very easily. They can move around in many animals. Three, they have interesting optics. Having a curved photoreceptor brings into focus things at a constant distance, unlike a planar one, which would bring in, as you have in your camera. So it's typical in biology to have things happen for multiple good reasons all at the same time. And that's what makes, so the problem is, which are the hypotheses that make this grid run? Well, certainly one is control over development. You just want to have developmental control over where you place things. Is that the whole story? Undoubtedly not. One of the interesting, I guess that my real question is, somebody was asking me yesterday about waveguides. The question is the brain like a waveguide, and if so, which kind? Does this grid structure have any functional significance? I really have no idea. One clue, well, we could talk about this for we should have a workshop where we, disappear into a pleasant location for 3 days and, download, the 15 key papers on it and then write a position paper saying, here's the evidence, we know it's genomic, we know it's developmental, we know it's evolutionary, but on top of that, the grid structure got doubled down in the forebrain, in the white matter, for the following 3 hypothetical reasons. We got to figure out which. Okay, so you say, yeah, there's all these reasons, but maybe there's a functional explanation. I like this one. I just try this in Manhattan sometime. It's just the observation that if you go northeast, if you want to go from here to there, you could have a group of friends who part company over here, and as long as they keep walking east and north, they'll get to that point in the same time. Now, I think I don't have to convince this audience that timing in the brain is sort of what the brain is about. And so I do find it intriguing that this structure has exceptional properties as a waveguide to maintain temporal coherence across large distances. Just a thought. Anyway, it's been a pleasure to have your ear for an hour, and pizza deliveries in the Upper East Side. Thanks for your attention.